Thank you so much. It was a fantastic first session this morning, or first two sessions, I'm not sure. And it's a pleasure to be here, and I'm really sad that I'm only here for the conference and not for a longer amount of time, because all of the posts on Blue Sky and what I've heard so far, it looks like it's a lot of fun and very productive. So I want to tell you about two vignettes, where the second I'll keep much shorter, and let's see how far I get. And I want to make a case for emergent properties of condensates, and that we have to be careful about how to think about them, and seeing is not, or seeing shouldn't always be believing. It is useful to use quantitative biophysical measurements and theory and computation. We, of course, have already talked about what condensates are, right? But you know, how one way of thinking about them is that they are not just large protein complexes, right? They are non-stoichiometric assemblies that can become really large, and they still preserve their high internal dynamics, at least in some instantiations of condensates. You can see this here by FRAP, right? These are stress granules. We've already seen stress granules. How does that work? And why might that be useful, right? So if we have proteins as examples of associative polymers that are multivalent for each other, then these can make clusters in subsaturated solutions. Sam has talked about different types of clusters in subsaturated solutions quite a bit, and gave a wonderful introduction therefore for you, so you have thought about this already. And you know, depending on the valence of these molecules that interact with each other here and their affinities, this will shape what this size distribution looks like, right? But basically, you'll have a lot of small stuff and a little bit of large stuff in subsaturated solution. And if we say that the large stuff is what you want, and because that has some sort of emergent properties, it's pretty difficult to make more of it, right? You can increase the concentration of the proteins here, and you'll get a little bit more of the large stuff and a little bit less of the small stuff, but it's really hard to make a lot of it. But if you then increase the concentration more and you know, make very large things, then you often also cross the saturation concentration, where now suddenly you have access to really large things. Because the way we look at it, you couple phase separation and percolation with each other, and now you have this percolated network of things within the condensates, and that gives rise to complexes here that are still dynamic, but you can populate these really large assemblies with interesting emergent properties. And of course, we have lots of them in the cell. I can jump over this. But basically, an idea is that these condensates generate specific internal environments that can drive or suppress biochemical functions. But it's not necessarily so clear whether they'll drive or suppress, and just because there is an enhanced concentration, it may not be so obvious, and you have to think about that a little bit more or measure it. Emergent properties of condensates could be, you know, that they're percolated interiors that give rise to long dwell times or lifetimes of interactions. They're viscoelastic. They have an interior network structure. So also conformations could be different inside than outside. The interfaces have specific structures and biochemical activities. There can be differential ion partitioning and interphase electric potentials, pH gradients over interfaces. And then, of course, this is not really the emergent property of the condensate itself, but there are also these clusters in the dilute phase. So if we understand these kinds of properties, does that help us understand how they may mediate biological functions and or disease processes? And I want to talk to you about two problems: the relationship of protein fibril formation to condensates in neurodegenerative diseases, and the role of transcription factor phase separation in transcription. Okay, we've done a lot of work on this protein, HNRNP-A1. Our original interest really came from the fact that mutations in this intrinsically disordered prion-like low complexity domain, long name for this part of the molecule, that mutations in this can result in familial forms of neurodegenerative diseases. But then we've also used it as a model system for understanding phase separation. And It can phase separate by itself, and it has this interesting primary sequence where we have lots of small polar residues and aromatic residues basically uniformly distributed throughout the sequence. And just to summarize how we think about how phase separation is encoded in this protein, we've identified aromatic residues, and of course lots of other people have too, but we've done this in a way that we understand what the interaction strengths are of these aromatic residues in the sequence. So we think the aromatic residues in the sequence provide the strongest interaction strength, and we describe them as stickers. And tyrosines are stronger than phenylalanines, and arginines can contribute to these interactions, and lysines interfere with them. And then residues in between, they mostly modulate solubility. If they're really small and don't like to interact so much with solvent, then they can, they can potentiate phase separation. If they're very large and really like to interact with the solvent, then they can suppress this phase separation. And then the net charge per residue of the protein also plays a big role in determining how strongly it phase separates. So we look at networking and solubility basically as two different things, and that allows us to put these kinds of systems on the same, into the same framework as multivalent domain motif type systems, like multi-SH3 domain, multi-proline rich motif type systems. Oh yeah, and of course, this also means not all IDPs phase separate, right? Because some may be highly soluble. Okay, how do condensates influence protein fibril formation? From work from Simon's and Tony's labs, and Mike Rosen and Roy Parker and mine and Paul Taylor, and lots of other work that came around the same time or afterwards, it was pretty clear that condensates have some influence on fibril formation. And the current view in the field, I would say, is that condensates promote fibril formation. And the hypothesis really was that came out of this work was that the high local concentrations in condensates promote nucleation, and therefore one gets faster phase separation. And this is important to understand because basically we want to know whether, we want to, what we want to do with stress granules with respect to neurodegenerative diseases, whether we want to stabilize them or disassemble them to prevent or treat these neurodegenerative diseases. And it may also help us understand how material properties impact biochemical processes. And this link between stress granules and neurodegenerative diseases comes from the following kind of observations. One hallmark of these diseases is that there are pathological fibrillar inclusions in the cells of patients, for example, TDP43, but also other RNA binding proteins. And these are typically stress granule proteins that fibrilize there. And then there are these whole bunch of RNA binding proteins that are found in stress granules when stress granules form, where recurrent mutations in these proteins lead to familial forms of these diseases. And then there is cell biological data where if you express, for example, DR1 is one of the RNA binding proteins that go into stress granules. This is relatively small here. I hope you can see something, but basically what you see if we express wild type protein and we form stress granules and then release the stress and stress granules disassemble again normally. But if you express disease mutants of this protein, then the stress granules disassemble more slowly and less completely. And this is a disease phenotype that is characteristic of these proteins. And then there's also a mechanistic link, right? And I'll show you here the one that was in our paper in 2015, where we looked at HNRNPA1, one of these RNA binding proteins in stress granules, where my colleague Paul Taylor had found mutations in its low complexity domain that drive these neurodegenerative diseases. And we could see that if we kept the protein in the one phase regime the whole time, we didn't see any fibrils over the course of the experiment. But whenever we dipped the protein solution into the two-phase regime, so at these points, then we could see condensates floating in solution. On the surface of the glass slides, we would shortly after that see fibrils forming, while without phase separation, there were no fibrils on the same time scale, right? So there's this idea that once you induce phase separation, you get fibrils earlier. Okay, and then the question is this a correlation? So this, I mean, the stress granules and the fact that there's fibril formation of stress granule proteins, or are stress granules really crucibles for neurodegenerative diseases? We've seen really nice work from Simon today, and there's also work from others, for example, Jim Shorter. And then there's the other side, like Don Cleveland, who have published work that suggests that stress granules are not the problem, and whenever proteins or TDP43 is not in stress granules, but in some other condensate, then that may be the problem. And perhaps that's also what Simon showed with the demixing. So how do condensates influence fibril formation? We're looking at wild-type protein versus disease mutants, and I should say we're just looking at the low complexity domain. This is not even full-length protein, but we're looking at a simple system to understand how condensates influence fibril formation. And we see condensates when we first induce phase separation, and then over time we see these things that contain fibrils. And here we see fibrils earlier in the presence of disease mutants, right? And the question is whether the condensates are required to form fibrils and whether they make this easier. And so, but instead of this kind of picture where we see condensates are on pathway to fibrils, I'll tell you now what our conclusions are. We found that condensates are off-pathway metastable states. They're basically sinks for soluble protein, and the protein had to flow out of these condensates to then, from the dilute phase, form fibrils, with some additional things that the interfaces of the condensates are important for nucleating fibrils. Okay? So now we're going to walk through some data to convince you that this is the picture that we're getting to. So we can measure the stability of the fibrils versus the condensates simply by looking in equilibrium how much of the protein is left over in the dilute phase. And we can see that the saturation concentration for fibril formation here is much lower than for condensate formation. Here it's in the hundreds versus 75 to 120 versus 1 to 0.6, right? So the fibrils are much more stable than the condensates. What we also see is that the disease mutations stabilize the fibrils and destabilize the condensates, right? Which tells you right away that it's not the same interactions that drive the formation of fibrils versus condensates. Probably not a surprise for people here in the room, but there's discussion about this in the literature, right? And so we have this picture, right, where the condensates are metastable relative to the fibrils, and we see that so there is this free energy gap between these two different states, and that free energy gap is larger for the mutant because the condensates are destabilized and the fibrils are stabilized by the mutation. Right? So how does that affect the kinetics of fibril formation? We can look at this by basically a thioflavin T fluorescence as a readout. Thioflavin T binds to the amyloid fibrils and has higher fluorescence in that state. And so we can look at these thioflavin T curves as a function of time to read out how much amyloid fibril is being formed. We do this here as a function of protein concentration. Everything that's blue is lower concentration below the saturation concentration. And then when we get into the red and the broken lines, then we're above the saturation concentration where we have condensates present. Okay? And we see these sigmoidal curves that are typical for nucleation and growth kind of kinetics. And we see that when we increase the protein concentration, we get shorter and shorter lag times, as you would expect, and larger slopes because growth gets faster, right? But it's kind of surprising that we get still shorter and shorter lag times once we hit the saturation concentration, because in this, here one protein, the dilute phase concentration should be constant once we are above the saturation concentration, right? And the dense phase concentration also. So no matter where the fibrils form, we wouldn't really expect that the lag phase keeps on getting shorter. And so this pointed to the influence of the interfaces. This had been seen before by Paola Rosi's lab and Evan Sproat's lab, and there are a number of other labs who have reported this now, and we see this also, where we can see that over time, we get enhanced thioflavin T fluorescence at the interfaces of condensates, suggesting that this is where nucleation happens. If we look now at wild-type condensates here on the right, then we see, again, that with increasing protein concentration, we get lower and lower, shorter and shorter lag phases, again, suggesting that, or showing, of course, this gets higher with increased concentration, but the lag phases keep getting smaller, even when we are above the saturation concentration, implicating the interfaces again. But what we then see when we look at these slopes here is that they're actually not getting higher. Perhaps a little bit here at the very beginning, but then they actually level off, and they are getting, the slopes are lower at the highest concentration than here at the lower concentration. And that suggests that really these condensates actually suppress fibril growth, right? They're not driving fibril growth, they're suppressing fibril growth. And Mina Farag and Kirsten Ruff and Rohit Papu's lab modeled this, and the simplest possible model was a three-phase model where we have a dilute phase, a dense phase with exchange between them, and a fibrillar phase. And when the fibrillar phase can only emerge from the dilute phase, then that was enough to model all of our data. It doesn't say that that's the only possible way to modeling it, but it was the simplest model that could explain our data. And we're using, primary nucleation, secondary nucleation, elongation, modeled on previous work from Thomas Knowles and Sarah Lindsay and others. And so this looks relatively complex. All I want you to take from it is that we can look at all of our different mutants. We can take curves below the saturation concentrations and above the saturation concentration. For the wild type, above the saturation concentration, we get these bimodal curves here. And with this simple model, we can explain the data. Right? And what we see when we extract our parameters from these fits is that the efflux out of the dense phase, so out versus into the dense phase, is much slower for wild-type protein than for the mutant proteins. And basically, the suggestion was that because of the higher metastability of the wild-type condensates versus the disease-mutant condensates, the efflux could be slowed down by the wild-type condensates. And that would make the wild-type condensate sinks for soluble protein. In principle, also these disease-mutant condensates, just that they are so weak sinks that we can't see a signal for that in our experimental data. And I'll show you a movie of this is for disease-mutant, I think, how this looks, just so that you get an image. So we're staining with thioflavin T, and you see some fibrils appearing, and then you see when the fibrils really grow, this is basically here bundles of fibrils that are forming. You see the condensates dissolving at the same time. Oops, I'll play this again. So when you see these fibrils really starting to grow, then you see these condensates dissolving, right? And so overall, our data so far suggests that, okay, the interfaces of condensates enhance nucleation of fibrils. The interior of the condensates can suppress fibril formation, and fibril growth happens in the dilute phase. And if we have condensates with higher metastability relative to the fibrils, so they're not as stable as the fibrils, but they're relatively stable relative to the fibrils, then the efflux would be slower, and therefore they would be better able to suppress fibril formation. And if that is true, then we should be able to use this to suppress the disease mutant phenotype, right? So now let's use our disease mutant that fibrillizes really strongly and stabilize those condensates more, make them more metastable. And we did this by putting tryptophan residues instead of phenylalanine and tyrosine residues in the A1-LCD. This really stabilizes the condensates as expected, right? It doesn't do much to the fibril stability. See here, the all W and the wild type are the same. In the disease mutant background, it's a little less stable. But overall, this really means that this delta G gap, this free energy gap, is now much smaller when we have the tryptophan residues in there. In this picture that I showed you earlier, right? Now the condensates are much more metastable relative to the fibrils than in the wild type or disease mutant case. And indeed, this really helps to suppress fibril formation. Now we're looking again at bioflavin T curves. These are for the disease mutant, and we knew that they grow very fast. The fibrils for the disease mutant grow very fast. But if we do this in the all W background, right, where the stabilities of the fibrils are basically the same as for the D262V alone, we can very much suppress fibril formation. And we can do this in trans also. So we have constant concentration of the disease mutant protein, and now we're adding this variant with the tryptophan residues. This has the disease mutation, and so the steric zipper motif that is the strong motif that seems to determine the stability of the fibrils. We have more in this now because we're titrating this in, but we can slow down fibril formation. And this kind of kinetic suppression was also recently reported by Xavi Sabatella's lab for CPEP4. There was this suggestion from our from the modeling that the condensates with high metastability mediate slow protein efflux, right? And our reviewers asked us to show whether this is really true. This was an important part of our model. And so Priya Banerjee's lab came up with this really cool assay where they're using a microfluidic chamber to capture individual condensates with an optical trap and then move them into a channel with buffer only and then look at their dissolution over time. Okay? And when we did this with wild-type protein, wild-type condensates, then they dissolve relatively quickly. And I want to mention this is under flux, right? So this dissolves much more quickly than if you are in equilibrium with the dilute phase concentration and you don't have any flux there. So they dissolve relatively quickly. Unfortunately, we couldn't do the experiments with the disease mutants because they fibrillized and they clogged up the microfluidic channel. When I asked whether they could try to do this again, they said no. The graduate student was scrubbing the device for 3 days. So, but what was possible was to do this in the all W background, right? And then the efflux becomes much, much slower. Makes sense, right? And luckily, we were also able to do this in the all W with the disease mutation. And there it's faster relative to the all W, really suggesting that the disease mutation destabilizes the condensate somewhat and makes the efflux faster. And overall, if we stabilize the condensates, we can make the efflux much slower, right? Really suggesting that this is a kinetic suppression of fibril formation in these metastable condensates. Does this say anything for the situation in cell and for stress granules? I don't really know, right? But we can try and do some tests. So Simon told you about the RNA binding proteins and stress granules and G3BP being an important RNA binding protein, like a scaffold protein together with mRNA to form stress granules. But there are lots of proteins with prion-like domains in this. We think of there being a strong prion-like domain network inside of the stress granules. And the question is whether, if we stabilize this prion-like domain network further, whether we can suppress a disease mutant phenotype. And the disease mutant phenotype that we're using is this ability to disassemble stress granules. I want to emphasize that this is an active process. This is not the same dissolution of the condensates that we're seeing in vitro. We're using this as a phenotype here, where if we're expressing disease mutants of an RNA binding proteins, the disassembly becomes slower, and we want to see whether we can rescue this. Okay? And this is here a quantification for this TR1 case. And then this also works for HNRNPA1, where the disease mutant, expression of the disease mutant slows down the stress granule disassembly. This effect is smaller than for TR1 because there's less HNRNPA1 in these stress granules. And this is what we then did. We used, of course, now here full-length protein. and express GFP as a separate polypeptide in the cell to mark cells with expression of H&R and PA1, comparing wild type, the disease mutant, and here the disease mutant where we put the tryptophan residues into the protein. And we can see that, as reported previously, the disease mutant protein, expression of the disease mutant protein slows down the disassembly of the stress granules. But when we put in the tryptophan residues, we can rescue the effect. We can actually over-rescue it. And this may come from different effects than in vitro, but at least it works. And so, here, seeing can be believing, but here in this case, perhaps there was some deceiving there as well, right? It's very clear from these kind of images that condensates are somehow involved in fibril formation. But in this case, there are two roles, right? There's nucleation at interfaces and suppression of fibril formation from metastable condensates. And these kinds of mechanistic insights from careful biophysical characterization and modeling really helps at getting at these questions. And now we can ask deeper questions about whether the stress granule network, if we stabilize that, whether that will be protective against neurodegenerative diseases. And I don't have much time left, but I'll try to very quickly make a case for how to look at this for transcription. Here, the challenge is that we have associative polymers, as we discussed, right? Then they form these soluble higher order complexes or pre-percolation clusters. And it's perhaps difficult to dissociate the function from these clusters versus the condensates. There were some papers with clusters. So in terms of transcription, we have interactions between transcription factors and co-activators, and they are multivalent in nature. They mediate transcription initiation, and they can lead to the formation of condensates, and they can lead to the formation of clusters or soluble complexes. So we use the case of GCN4, a yeast transcription factor that has a lot of data on it, is very well characterized, and the interactions with its cognate mediator subunit MED15. And there are multiple domains in MED15 that interact with multiple motifs on GCN4. You see here that there are two transactivation domains, and we concentrate on one of them, but there are even in that second transactivation domain, there are multiple motifs that interact with these domains on MED15. And the problem, if we don't know whether the soluble complexes or the condensates mediate the function, right, then even if one sees condensates in cells, as in this paper, one doesn't know that the activity is actually coming from the condensates. There's a lot of stuff in the dilute phase that one doesn't see by microscopy, right? So we decided to tackle this together with Asim Ansari at St. Jude, who's an expert in transcription, has worked on GCN4, early on in his career and has a lot of knowledge about this. Anna Bremer, when she was in my lab, tackled this, and Walter from Asim's lab. And how we decided to do this was to make lots of variants of GCN4, measure activities in cell, in vivo, in yeast, and compare this with affinities for MED15 of the GCN4 variants, and the abilities to phase separate. And this is, in this case, it's heterotypic phase separation between multiple components, because it's all about recruiting mediator, right? And I'm not going to go into much detail of how we did this. Tell you the message briefly. Formation of complexes and condensates can explain the activities similarly well. It's really hard to use these mutations to decide where the function lies. But there were some variants for which the condensates explain the activities better, but then they seem to dampen activities. Condensates can carry out function, but perhaps they actually dampen them relative to complexes. So very briefly, I'm just showing you that we made a bunch of variants and that we had a nice range of activities. The wild type lies here. So we can read this out pretty well. And this is the data, and there's a lot of data, but I'll just want to show you what this means. So here we're plotting the formation of heterotypic condensates versus activity. Here, the formation of complexes, soluble complexes versus activity. And here we're comparing the two biophysical readouts. And overall, what we see is that, while they're scattered in the data, overall, we can, but both of these biophysical readouts explain these data relatively well, right? So first message is the simple in vitro reconstitutions can be good reporters of in vivo activities if you reconstitute the relevant interactions, right? Then the two biophysical readouts are really highly correlated, which means you can't really use these mutations very easily to tell you whether the condensates are where the function is coming from, right? At least you need to compare this to a complex model. But there should be partial separation of function mutations, because if we think about phase separation as phase separation coupled to percolation, right? So if we just make complexes, and this would be very high concentrations of the proteins, the multivalent proteins without phase separation, right? Just multivalency doesn't give you a second phase. You need this density transition that comes from a relatively low solubility. I just have like one more slide. Relatively low solubility to get that density transition. So it should in principle be possible to make at least partial separation of function mutants. We tried to do this, but we weren't very successful. But perhaps we have some variants where we saw something like this, right? So if we look here at these red variants, these are the ones in the two biophysical readouts that are not as well correlated as the others. And if we look at these here in our experiments versus activity, they fall very well on the line for the condensate readout, except for this one variant over here, but not so much for the small complex readout, right? And that suggests that the phase separation may explain the activities better. But if we look what this means here, you can see actually that the KD axis is reversed. So up here, these that fall off this correlation, they have very high affinities, but they don't deliver as high an activity as their affinities are. So that suggests that they probably phase separate because they fit well here, but then their the interconversion or the activity that they deliver is not as good as it could be. And so their emergent properties, perhaps their viscosities, viscoelasticities, don't allow for the full activity that they might have. So transcription factor phase separation seems to have a duality of roles. These condensates can probably function equally well to complexes most of the time, unless they have very high affinities, and then they may actually dampen the activity. But of course, that doesn't mean that phase separation in transcription isn't important. There's probably roles for coactivator, mediator, pol-2, condensate formation. There's really nice work from Ibrahim Sisse and Ben Sabari on this. But you have to be careful when you make these mutations to dissociate where the function is coming from, right? And so we have these emergent properties. We have to remember that the high local concentration does not necessarily have to be the dominant effect on biochemical processes, kinetics versus thermodynamics, right? There's network structure inside them that could either suppress or activate biochemical processes. There's also differential partitioning that can play roles and so on. With that, I thank people in the lab who did the work, wonderful collaborators and funding. I'd be happy to answer some questions.
Tanja, that is beautiful as always. I had not actually seen the first story. That is so clarifying as to what is going on. It's really, really beautiful stuff. I want to ask this in the right way because I really mean it, and I don't want to get shoved into a semantic discussion. We spent so long distinguishing between phase separation and they kind of invented biomolecular condensates because we realized the world was bigger than just that process. And you use this word cluster versus condensate in a way that is clearly very meaningful. And so I want to kind of say, how should we think about the differences between these two phenomena, given that we're no longer talking about phase separation necessarily?
Right. So, right, in the first part, I wasn't really talking about clusters. I was talking about condensates. But in the second part, I was clearly talking about the difference between the two things. And I don't have a good answer of how to addressing this once the clusters get really high and where is the boundary, right? Clearly, it's not just a size because that is system-dependent size where it's transitions from a cluster to a condensate. Perhaps it's, perhaps it has to do with having a full interface and what is the size at which you can first make this full interface. Perhaps it's not so important, and the theorists, I would welcome them to chime in, because as far as I understand, once you get to really large clusters, they start to have these emergent properties that the condensates have also, and that is basically a lot of them scale with size. It's just really difficult to make very large clusters for a lot of cases. And once you couple the networking to the density transition, now you can make these really large assemblies. And so that, there's probably some blurring between these things and how they act, especially in cells where you don't have super large systems, right? So, that would be my,
thank you.
my
yeah, thanks a lot.
Yeah, Tanja, so this all W mutant, the effect is really cool. And I'm just wondering, the effect in vitro and in cells, could it also be different reasons? I mean, because I'm asking because for TDP and for FAS, we've seen that RNA binding is very often the key thing. And I'm wondering whether the all W mutant holds on to RNA more tightly maybe, or is there something about RNA binding that may also be playing a role in cells?
It's certainly possible. We haven't looked at binding of the all W to the RNA, but my expectation would be that it would bind to RNA more strongly. So that's why I said, right, I don't know whether it's for the same reasons. Clearly, there are more experiments in cells that we need to do, right, with more subtle mutations than putting in a lot of tryptophan residues. It's a very good point, right? I don't want to I don't want anyone to take away that we know for sure that the stress granules suppress fibril formation. It's our model based on a very simple in vitro system, and clearly we need to make that in vitro system more complex. You showed really nice data that perhaps for TDP-43 something different is going on. I just also want to point to the possibility, right, that when in your TDP-43 system, perhaps these demixed states at the beginning, they may come from the dilute phase, as you also pointed out, right? And I think your data very strongly says that it comes from the oxidative conversion to disulfides that you get this demixing. It is not because of the high concentration in the dense phase. And what's going on in the dilute phase, you may have the data, but I don't see what's going on in the dilute phase in your case, right?
Excuse me, you very nicely distinguish between, in the in vitro experiments, between the interior of the condensate that protects against fibril formation and the surface of the condensate that nucleates. And nucleates is a very general term, and I was wondering if our idea that the proteins at the interface of a condensate and an aqueous phase might be in an oriented amphiphilic configuration might be related to this nucleation. And we know from other physical systems, when you have a molecule that has, an A part and a B part, it can very often assemble to make chains. So the fuzzy picture I have in my head is that your condensate has proteins in the amphiphilic conformation at the interface, but also some proteins with that conformation inside. And that's all metastable, let's say. And those proteins at the interface then start to zip up because they're oriented to make fibrils and maybe grab more of those oriented proteins, non-oriented but still amphiphilic conformations inside. And how would one know that without looking in detail? Maybe one should look at correlations between fibril formation at the interface and the interfacial tension of the condensates, where you could vary things, not just no fibrils to fibrils, but where you could look more gradually.
Right. Okay. I have a couple of thoughts on this, and then, we'd love to do those experiments that you're suggesting. So one thought I have is that this is a case. It's probably not so helpful for you to see the sequence, but I can show you the sequence later, and you could do your own sequence analyses. This is a pretty well-mixed polymer, in the sense that we don't have any parts that are more charged than others, or where the aromatic residues are on one side versus more hydrophilics on the other one. I think this is one of the more well-mixed heteropolymers in that sense. So we look at it more as, while we know obviously it's a heteropolymer, and there are these aromatic residues that give rise to stronger interactions than other parts, it effectively behaves like a homopolymer, is the way
That's your intuition.
Pardon me?
That's your intuition.
That's what we have, that's what our characterizations show with Rohit Papu, for example, in our 2020 paper.
So if you were to do EPR on that, you would see one signal per polymer, per protein. Each protein would have one average rotational diffusion constant, and okay, I'm not questioning you, please.
No. I don't know. We haven't.
Okay,
PR.
that might be. I didn't say that every protein should do that.
Absolutely.
I'm just saying that tendency can lead to ordering.
Right. But I have a couple more thoughts. So just EPR, we haven't done on this, but NMR spectroscopy, for example, gives rise to a very nice disordered system. We know actually by single molecule FRET that in condensates we get at least two populations. Okay, yeah. Yeah, but I don't think it's the interface. We don't have enough signal to see any difference between the interface and the inside. From simulations of the system where the parameterization directly comes out of our experimental data, we know that there is some that there are extended conformations of parts of the chain at the interface, and that they are perpendicular to the interface. So that's what you're suggesting is exactly what we're seeing in these computations, and that's also what we think might be what's driving the nucleation. And another point there is that once you go to a really strong driving force for phase separation, other computations from Creston and Larson's lab suggest that now they are the chains should not be perpendicular, but now horizontal with the interface. So the orientation changes with the driving force for phase separation. And indeed, once we go to the all W variant, we don't think that we have strong nucleation at the interfaces. So all of that would fit into that picture.
So the difference between what I suggested is whether they're just stretched out at the interface but act like homopolymers, or have some dual chemical nature that might make them chain. Right. Chaining usually, in many systems we have a sort of, not electrostatic, but magnetics, plus minus, you get these chains, plus minus, plus minus.
Interesting.
Okay, thanks.
Hey, Tanja, that was really wonderful. I think it was pretty clear. I just wanted to make sure, but in the model for the rates of aggregation, you have no rate of interconversion. You're saying all of the aggregation happens in the dilute phase. There's no rates for within the condensate. Okay.
We don't have any sign of Fribble formation in the dense phase. And we've done this for a bunch of different prion-like domains. We don't see any sign of Fribble formation on the inside. When we look at other people's data in the literature, we don't see any signs of Fribble formation on the inside of condensates.
Thank you.
Last question from Will.
Sorry, I had basically the same question. I just wanted to maybe ask one slightly different aspect. Just point of clarification. When you were describing the lag phase, I thought you said something about this suggesting that the interface is involved. Could you just clarify how that relates to this model that says that you only need primary and secondary nucleation in the, primary and secondary nucleation in the dilute phase? Thanks.
Yeah. The simple chemical kinetics model doesn't take the interface into account, but we can model things with just, so we're not modeling our whole concentration series together. So this is probably where some of that information gets lost, where we would need to invoke the interface specifically in the chemical kinetics. So our data where the lag phase becomes shorter even when we are already supersaturated with respect to condensates suggests that the interface is likely involved. And we see this bioflavin T signal at the interface, but then we're not carrying it all the way through in the chemical kinetics modeling. So basically we don't address it in the chemical kinetics modeling. That's all I can say about that.
Okay, so then let's thank Tanja and all the speakers today.